From our lessons on the developmental transition and flowering process in plants, we will now begin our lecture on seed formation and development, which is covered in Module 2 of our syllabus. In this module, we will cover the various physiological processes that occur within the flower towards the development of a seed. For today, our focus will be on gamete formation in plants, specifically male gametogenesis. Reproductive stage of plant development can be divided into gametogenesis, embryogenesis, and endosperm development. Our focus today will be on gametogenesis or gamete formation. We've already established the fact that plants develop new structures or organs when they transition into the reproductive phase, and the key organ that develops during this phase is the flower. Flowers are the main organs involved in the sexual reproduction of plants. Recall that flowers have four basic parts or organs arranged in concentric circular whorl. These organs include the sepals, petals, stamen, and the carpel. Among these, only the stamen and the carpel are considered reproductive structures. The stamen is the male reproductive organ, while the carpel is the female reproductive organ. Within the stamen and the carpel, the male and female gametes respectively are formed. So what are gametes? Gamete is a general term that refers to the male and female reproductive cells. Specifically, the male gamete is known as the sperm, whereas the female gamete is known as the egg. The union of these cells during fertilization following pollination results in the formation of a zygote. Before fertilization can occur, a flower must first produce the sperm and the egg. In flowers, the sperm cell is produced in the pollen grain. The structure or organ that produces the male gamete is known as the male gametophyte. Since the pollen grain produces the male gametes, it is considered the male gametophyte. Pollens, in turn, are produced in the anthers of the stamen. The egg cells are produced in the embryo sac. The structure or organ that produces the female gametes is called the female gametophyte. The embryo sac produces the female gametes and is therefore the female gametophyte. The embryo sac is produced within an ovule in the ovary of a carpel. This slide shows you an overview of gametogenesis in flowering plants. The term gametogenesis just means gamete formation. The top panel illustrates the process of male gametogenesis, while the lower panel illustrates the process of female gametogenesis. Both male and female gametogenesis can be divided into two events, namely sporogenesis and gametogenesis. We will discuss this in more detail later, but at this point, I want to make the distinction in the terminologies used to describe these processes in the male and female gametophyte. Notice the use of micro to describe these two processes in the male gametophyte, microsporogenesis and microgametogenesis. In the female gametophyte, the prefix mega is used. We have here megasporogenesis and megagametogenesis. The use of these prefixes is in reference to the size of the male and female gametes. Sperm cells are 1,000 times smaller than the egg cell, hence the use of the prefix micro. Because egg cells are relatively way bigger than the sperm, the prefix mega is used. At the molecular level, more is known about pollen grain than embryo sac formation, and there are two main reasons for this. First, the flower produces more pollen than embryo sac. A single corn plant, for example, can produce more than 25 million pollen grains, but only a few hundred embryo sacs. That's why a single corn plant produces only 1 or 2 cobs with 200 to 400 kernels. The point is that, since pollen grains are produced in higher numbers, there are far more specimens that can be examined. Secondly, pollen grains are released from the anthers, so it is easy to isolate and study them. In contrast, to get an embryo sac from a flower, you should first have to dissect the flower and then remove the ovule tissue to isolate the embryo sac. Let's now take a detailed look into the series of events that occur during male gametogenesis or male gamete formation. In general, male gametogenesis is highly conserved across plant species. In this top left corner is an illustration of an anther. The series of events that leads to the formation of sperm cells happens within the anther. If we cut across the anther, we will see a cross-section that looks something like this. During the early development of the anther, diploid cells will divide to form microspore mother cells and tapetal initial cells. The microspore mother cells will be contained in cavities known as the pollen sacs. Those are the 4 pockets you see in the anther cross-section. It's right here. Both the pollen mother cells and the tapetal initial cells have their own functions and contribute differently to the production of pollen grains. Let's first take a look at what happens to the microspore mother cell. As I said, microspore mother cells are produced from diploid cells in the anther. What do we mean by diploid? A diploid cell is a cell that contains 2 complete sets of chromosomes. Each microspore mother cell will undergo meiosis or reproductive cell division to produce 4 haploid cells. What do we mean by haploid cells? Haploid cells are cells that contain only a single set of unpaired chromosomes. Each of these haploid cells is known as a microspore. Collectively, they are called tetrad because they come in groups of 4s. Each tetrad is enclosed in a callose wall, just like what you see here, and that callose wall is made up of polysaccharide callose beta-1,3 linked glucan. Now, for each of these microspore comprising the tetrad to develop into a pollen, they first need to be released from the callose wall. For this, we need the enzyme callase. Callase is an enzyme secreted by the tapetum. The tapetum is the wall lining the pollen sac. More on that later. Now, for now, I want you to remember that the whole process that we just described, beginning from the development of the 4 haploid cells from the microspore mother cell via meiosis, up to the release of these 4 microspores from the callose wall, as you can see right here, through the function of the callase enzyme, is called microsporogenesis. So what happens when microspores are released from the callose wall? Each of the microspores undergoes an asymmetric cell division to produce a pollen grain. We say asymmetric because the product of the cell division is a pollen grain with 2 cells that are unequal in size. The larger cell is the vegetative or tube cell, and the smaller cell is known as the generative cell. Subsequently, the generative cell divides to produce 2 sperm cells. The timing of this division varies across plant species. In Arabidopsis, for example, it occurs while the pollen is still on the anther. In the majority of angiosperms or flowering plants, the pollen grain is shed from the anther while it is still at the 2-cell stage when you have the vegetative and the undivided generative cell. Division of the generative cell takes place only after pollination when pollen tube growth begins. In either case, the final product is a pollen grain consisting of 2 sperm cells surrounded by the cytoplasm of the vegetative cell. Pollen grains then undergo a controlled process of desiccation that brings it to full maturity. The controlled desiccation process serves to arrest the metabolic processes in the cell until such time that it is ready for pollination. The whole process that begins with the asymmetric division of the free microspores up to the division of the generative cells to produce 2 sperm cells and pollen grain dehydration is called microgametogenesis. What about the vegetative or tube cell? What is the function of this cell? The vegetative or tube cell is responsible for the accumulation of nutrient reserves and proteins required for pollen tube growth. During pollination, the tube cell leads the elongation of the pollen tube. We will talk more about the function of the vegetative or tube cell when we start our discussion on pollination and double fertilization in plants. A mature pollen coat is composed of a lipid protein matrix that protects the grain from excessive desiccation. Remember that for a pollen to mature, it needs to undergo a controlled process of dehydration. I emphasize the word controlled because excessive drying can kill the gametes. Again, the purpose of the drying process is to suspend the metabolic processes in the pollen until such time that it is released and lands on the stigma of the female flower and is ready for fertilization. The way of the plant to ensure that the pollen and ultimately the gametes don't die is to have a protective coating in the surface of the pollen itself. A closer look at the structure of the mature pollen coat will show an inner and outer wall. The inner wall is called the intine, which completely encloses the grain. The intine contains cellulose and arabinins, but the main component is callose. The outer wall is known as the exine. The exine has apertures at intervals in the form of ridges and spines. These apertures serve as sites for pollen tube germination. The exine wall of the pollen grain in many species shows an elaborate pattern of spines, ridges, and other decorations and is almost indestructible. To illustrate that variability, we have here a collection of pollen grains from different plant species showing elaborate apertures. We have here pollens from water lily, nettle, plantain, and so on and so forth. Because the patterns are often unique to particular species, they can be useful in identifying the plants associated with a particular location. This attribute has been applied both in paleobotany to determine which plant species occupied a habitat in the distant past, and also in forensic science to identify crime scenes. The extraordinary durability of pollen resides in the main structural component of the exine wall, the sporopollenin. This is a highly decay-resistant phenolic polymer that allows the exine to retain its structure for millions of years and also under very harsh conditions. Now let's not forget about the tapetal initial cells. We know that tapetal cells are produced from the division of the diploid cells in the anther along with the microspore mother cell. We already know what happens to the microspore mother cell. The question now is, what happens to tapetal initial cells? The tapetal initial cells will undergo mitotic divisions to form the tapetum. Now the tapetum is the tissue that lines the pollen sac in the anther. What are the functions of the tapetum? We already talked about one function of the tapetum earlier. For one, the tapetum produces the enzyme callase that is required to break down the callose wall surrounding the 4 microspores produced by the division of the microspore mother cell. The tapetum also produces structural polymers and pigments that make up the outer wall of the pollen. Late in pollen development, the cells of the tapetum disintegrates and their cytoplasmic content are deposited on the pollen coat. To summarize, the sexual reproduction of plants requires the development of gametes in the form of sperm and egg cells. Without the formation of both gametes, the plants will not be able to complete their life cycle. Gamete formation undergoes 2 distinct phases, sporogenesis and gametogenesis. In male gametogenesis, these 2 processes are specifically referred to as microsporogenesis and microgametogenesis in reference to the smaller size of the male gametes or the sperm compared to the egg cell. In flowering plants, the male gametophyte or the organ that produces the male gamete is the pollen grain. Pollen grains are produced in the anthers. The final products of male gametogenesis are 1 vegetative cell plus 2 sperm cells. The vegetative cell leads pollen tube elongation, while the generative cell divides to produce 2 sperm cells.